Hello, this is a recorded lecture for Chapter 7, The Blueprint of Life from DNA to Protein. And this is in the recorded series for Microbiology 230, and I am Dr. McGraw. This chapter is going to really look at the DNA modifications and the DNA structure that we can better understand how certain antibiotics and certain dysfunctions of an organism can go awry based on DNA. If you look at history in this, which is kind of important, is that in 1866, Gregor Mendel determined trace inherited as physical units, and they're now called genes. Gregor Mendel was actually a priest, and he did this study over a great deal of time with bean plants, strangely enough. In 1941, excuse me, George Beadle and Edward Tatum reported that genes direct production help produce the enzymes of the body, and they treated mold, Neospora species, with x-rays. They established metabolic defects inherited as a single gene, and they concluded a single gene determines the production of one enzyme, and they were awarded the Nobel Prize in 1958. So DNA is the blueprint of life, as we said before. It exhibits what's called incredible diversity of life determined by what's in the DNA. And strangely enough, there are 4 proteins that make up DNA, that make up the steps of DNA, and they're adenine, thymine, cytosine, and guanine. And interestingly enough, they can code for an enormous amount of information, and 3 nucleotides encode a specific amino acid. So 3 nucleotides together determine a protein, or amino acid is going to be created, and several amino acids make up a protein. So you see that the potential for diversity is unlimited, and you'll hear the term greater than a billion or infinite or whatever, but it really is pretty infinite. I'm not sure we've actually identified every single possible code we have based on the variations in these 4 little nucleotides that make up these multiple proteins. If you open your D2L module for this particular chapter, you'll see that there's a PowerPoint there, and you can open the PowerPoint up, download that PowerPoint, and then you can do a screen, what's called a slideshow, and that's for this. And then when you get for this particular lecture, and then you can click on this DNA structure video, and it'll kind of walk through that. There's no way to do that in this type of lecture, so this recorded lecture, so I have to have you do that manually. Now, a genome is a complete set of genetic information, and it's a chromosome plus technically some plasmids that go along with that. In all cells, with the DNA, if you will, and viruses actually have RNA. We know that some viruses are DNA, and a functional unit is a gene. So it encodes the gene product, usually a protein. Now, studying nucleotide sequences is called genomics. And so just terms we use as we go through this lecture. Now, cells must accomplish 2 tasks to multiply. They have to be able to replicate their DNA, and they have to show gene expression, that is transcription, translation. And you'll see this a lot, transcription, translation. It's interesting that what happens is the DNA, which is in the nucleus or nucleoid of the cell, has to send out messages to the rest of the cell. Turn up the heat, turn down the heat, make this enzyme, don't make this enzyme, that kind of thing. And so what happens is transcription is, if you think about what transcribers do, they write down things that people say. And so inside the nucleoid or the nucleus, there's a site where a DNA creates, if you will, an RNA, which is called messenger RNA. Think of messenger RNA as a knotted rope. It's very specific and causes a very specific thing to happen. And then what happens is this messenger RNA leaves the nucleoid or nucleus, depending on the cell we're talking about, and it goes out to the rest of the cell, primarily the ribosomes, and there the ribosomes translate that message and create the protein that's being directed by the nucleoid or nucleus. So the information flows from the DNA inside the nucleus or nucleoid to create RNA, and then ultimately the ribosomes, which you remember from previous lectures, I hope, create protein. And so that's kind of the central dogma of all biology, cellular biology, including the molecular biology. So DNA is, as I said, so what we're seeing here below in this graphic is the DNA is being replicated to create 2 new cells, which has to be replicated. But it also shows that gene expression as we go forward to create that protein that we need for whatever action we're trying to create. Now our DNA, the DNA forms a double-stranded helices as far as inside the human cell and many of the bacterial cells, if you will. And one of the contents, or one of the elements, is deoxyribonucleic acid, i.e. DNA. And so you get the idea. Now nucleotides are joined between what's called a 5-PO4 and the 3-OH, which is a place on the DNA molecule itself, on the DNA strand. So these are just locations. Think of them as GPSs. And this forms the sugar phosphate backbone. A single DNA strand will have a 5 and a 3 end. So I have 5 at one end and a 3 at the other. So that's kind of the destination. Think of this as a GPS at each end. One end has a GPS of 5' and the other has a GPS of 3'. And in a DNA that's a double helice strand, the 2 strands are parallel, but they're called anti-parallel. That means at one end is the 5 for one and the 3 for the other, and the opposite is true on the other end. One's a 5 and 3 in that sense. And so the strands are considered complementary but anti-parallel. They're held together by hydrogen bonds between the nucleobases, and there's a pair sharing. And the pair sharing, for instance, is adenine and thymine. It's attached to adenine and thymine for 2 hydrogen bonds, and cysteine and guanine is attached together with 3 hydrogen bonds. And separate strands are called melting or denaturing. So separating those strands to break them down is that melting or denaturing of the DNA double-stranded helices. So in this process, if you take that DNA we see here, and we look at that in a more geographical sense, you see the double strands, and you see the stair steps here. What you're seeing is the sugar deoxyribose, that is the sugar strand, and then you see the phosphate that field holding together. And here we see the exact same thing on the other side, if you will, but in opposite directions. So that's kind of a way to look at how the strands go together. Now RNA, ribonucleic acid, is a little different in the sense that ribose is used instead of deoxyribose. And then instead of thiamine, in the four elements we talked about, uracil replaces thiamine. It's usually a shorter single strand, and it's synthesized from a DNA template. It can be messenger RNA, the little m, like we talked about before. It can be transfer RNA, which is a translator RNA that the ribosome uses to translate the message from the messenger RNA. And then there's a ribosomal RNA that helps to produce the protein that we're talking about. So RNA molecule is a transcript, means it's transcribed from the DNA inside the nucleoid or nucleus, and then it goes to the cells themselves, or to the ribosomes themselves, where it's translated. And the base pairing rules apply except uracil pairs with the adenine, like we said before, and the transcript quickly separates from the DNA, so this is like a messenger. The ribonucleic acid has 3 types of required for gene expression, like I said. There's messenger RNA, which is little m, ribosomal RNA, and then transfer RNA. And you're seeing those different parts. So there's a transcription piece that comes from the original protein, and then we create those RNAs that we talked about. Now, cells control levels of protein synthesis. That's how they do that. And protein is the driver of everything. Protein could be in the form of a new enzyme, could be in the form of a substrate, if you will, to create a patch for the wall of the cell. It can be a substrate to form to replace one of the organelles or the small organelles, if you will, inside the cell. And there's rapid degradation of the messenger RNA transcripts are important. So they basically break down pretty quickly. So they're made as to be, their life expectancy is short because you don't want them floating around, causing other proteins to be created that you don't need. And so what happens is there's a reproduction is done again and again to get the same proteins. This is a protein synthesis video. Again, you could download the PowerPoint and you can use this. Look at this. Now, DNA replication is usually bidirectional. So we're going to replicate the DNA. The cell is going to multiply from 1 cell to 2 cell through binary fission. And what happens is the 5' and the 3' at one end, 5' and 3' at the other end. And so what happens is in this process of breaking down the DNA is that replication is what's called semi-conservative. So what happens is the two spirals, each spiral separates from the other spiral. Half goes one way, half goes the other. And so each of those pieces work as a template to build on a new DNA. And so that's called a semi-conservative method, so they're not building from scratch. And the reason that's important is it helps to reduce the potential for a mutation, for mistakes, as it were. So replication is semi-conservative. DNA contains one original, one newly synthesized strand, and replication begins the origin of the replication. So proteins recognize and bind to that origin of the replication, and they melt that double-stranded DNA. The primases synthesize short stretches of complementary RNA called primers as they build this molecule. So bidirectional DNA replication there's a good video located there, and I suggest that you download that. This is DNA replication. So we have that double-stranded DNA. It's then broken down into two single strands, if you will. And the single strands are then, what happens then is the single strands are then each then rebuilt into a double strand. So ultimately you end up with two identical DNAs to the original, the mother DNA, as it were, and the two daughter DNAs. And that's done by that technique we just talked about, that semi-conservative technique that we talked about. It's really important to remember that. It's also important to remember that as they are rebuilt in opposite ways, if you will. The one strand, or the A prime strand, if you will, is built in the direction of one from 1' to the other, and so 5' to 3', and the same thing happens to the others. Because the strands are anti-parallel, so to speak, then they are built in different directions. They go in different directions for the building part. This is talking about the DNA replication. This video is a good example of how it's put together. You want to take a look at that. So DNA replication, this shows the original DNA strand, and you see the replication fork. There's a fork that separates that DNA strand into two half parts, as it were, two half pieces. And then you see this, the replication forks are there to break them apart so that they can be separated and ultimately to be rebuilt into two full strands. So there's a structural basis for DNA replication. That's another video. Great video to download and take a look at. Now, DNA replication is a coordinated process. There's many enzymes and proteins. There's that are called replisomes, assembly line. Let's look at all the parts of that. And these are kind of the components of DNA replication bacteria. There's DNA gyrase, and that's an enzyme that temporarily breaks the strands of DNA to relieve the tension caused by the unwinding of the two strands of DNA helices, so they become two linear strands, not two circular or spiral strands. There's DNA ligase that joins two DNA fragments together by forming a covalent bond between the sugar and the phosphate residues of the adjacent nucleotide. There's DNA polymerase that synthesizes the DNA. They use one strand of DNA as a template. So the DNA polymerase will help to build that new strand. And then, as far as the helices are concerned, their origin of application is called, there's a distinct region of the DNA molecule which replication is initiated. The primase is an enzyme that synthesizes small fragments of RNA to serve as primers for DNA synthesis. I'm sorry, helicases are enzymes that unwind the DNA helix at the replication fork. The origin of the replication is distinct in each strand. And the primer is a fragment of nucleic acid which DNA polymerase can add nucleotides, and that's the process. They start that process and move through it. A replosome is the complex of enzymes and other proteins that synthesize that new DNA molecule. And the Agasaki fragment, that's a nucleic acid fragment that is produced during discontinuous symbiosis, the lagging strand of DNA. There's the lagging strand is the second strand of DNA, the one that's backwards or built in the other direction. So DNA polymerase is synthesized in 5 to 3 directions, as I said earlier. Hydrolysis of high-energy phosphate bond powers the DNA polymerase, and they can only add nucleotides. They're not initiated. So they require primers at the origin of replication, and this is the process they do, primers that sort of get things started, as it were. The helicases unzip the DNA strand. They basically unzip this so we have two halves instead of one whole. And there's the leading strand, that's the prime strand, that's the main strand, the prime strand, and the lagging strand is the other strand. They just call it that way. The leading strand is synthesized continuously. The lagging strand is synthesized in a discontinuous sort of manner, which is interesting. So the leading strand goes from A to Z. The lagging strand goes A, Z, or A, Q, R, Z, P, L. It goes in a discontinuous sort of way. The DNA polymerases can only add nucleotides to 3' end, and the production of Okazaki fragments occur, and different DNA polymerases replaces the primers. So DNA ligase forms this covalent bond between the adjacent molecules and rebuilds the new two DNAs that are being created from the original DNA. Replication produces 2 complete copies of DNA. Each daughter cell receives 1/2 of that, if you will. Replication, for instance, of E. coli chromosome takes about 40 minutes, but optimal generation time is about 20 minutes. A cell can initiate another round of replication before previous round is complete. So these are not linear; they can occur in a somewhat semi-parallel sort of way. Each daughter cell inherits 1 complete chromosome already undergoing replication. So it's kind of just an interesting process when you think about it. Now, transcription is the gene expression in bacteria. Transcription is really also in eukaryotic cells. The difference is in prokaryotic cells it occurs in nucleoid, and in eukaryotic cells it occurs in nucleus. So transcription, the RNA polymerases synthesize a single strand of RNA. So think of this as taking a message off the DNA blueprint to go out to the cell to cause something to happen. It uses the DNA template. It synthesizes in 5 to 3 direction, and it can be initiated without a primer. And it binds to what's called a promoter, which is found upstream on the genes, and it stops at a terminator. So promoter is kind of the start button, and the terminator is kind of the end button. And that's just a way to sort of think about these. So promoter is the start, beginning of what's going to become the RNA piece, and the terminator is going to be the end of that. This is a great look at messenger RNA synthesis, transcription. You'll want to take a look at this off the video, off the PowerPoint. Now, transcription RNA sequence is complementary, which means anti-parallel to the DNA template strand. So basically, the DNA template is a minus strand, and the complement is a plus strand. So it works in the opposite direction. So RNA has the same sequence as the positive DNA strand, except the uracil instead of thymine. So, like I said, we talked about this before. The uracil will replace thymine in the RNA messenger RNA. Messenger RNA transcripts are as a monocistronic, 1 gene. The polycistronic is multiple genes. You can see that as you go. And so that's the transcripts we're looking at. Now, the sigma factor recognizes the promoter, a subunit that's loosely attached to RNA polymerase, and there are various types that recognize different promoters, and so they synthesize controlled transcription of a set of genes. Eukaryotic cells and archaea use the transcription of factors, and this talks about the different strands of promoter of the RNA polymerase, the sigma factor, and the terminator that we just talked about. It's also located in your book at table 7.2. Transcription is, like I said, basically a reading process, and I don't really want to get too deep into this other than let you know that what's going to happen is there's a promoter that's going to help to sort of create a reading of this information, and then there's a terminator at the other end that says, okay, that's the end of the message, this particular message. There's initiation, which the RNA polymerase binds to the promoter and melts a short stretch of DNA. The elongation, the single factor association from RNA polymerase leading the core enzyme to complete transcription. RNA is synthesized in the 5' to 3' direction, 5' to 3' direction, as the enzyme adds nucleotides to create the message we talked about. And the termination is when the RNA polymerase encounters the terminator, and it falls the template. It falls off the template, and it releases the newly synthesized messenger RNA or RNA. So that it kind of creates this scraping, if you will, of the information. That's a good term. The promoter orients the direction of transcription, so that's the initial piece we talked about. It's found upstream of the genes. We talked about where that happens. Once the RNA polymerase is moved past, another RNA polymerase can then create the binding. It allows rapid and repeated transcription of a single gene. So this is RNA prime 3 to 5 and 5 to 3 in the opposite direction. You see that. Now, the process the process of decoding information of messenger RNA is translation, and this occurs not in the nucleoid or the nucleus as transcription does, but occurs at the ribosomal location. So this is a ribosomal function. Messenger RNA is a temporary copy of the genetic information. Major components are messenger RNA, the ribosomes, transfer RNA, and the associated proteins, as well as the ribosomal RNA. So in this process, the components of translation is an anticodon, messenger RNA, polysome, if you will, a reading frame, a ribosome, a ribosome binding site, the ribosomal RNA, a start codon, a stop codon, and the transfer RNA. So those are the components we're going to talk about in the translation of this information. There's a translation elongation, as it were. Again, I would strongly suggest that you download that, take a look at it. Now, I'm not going to go through and make you remember all these because that would just be mean, but I just want you to see the complexity of the process in that creating this genetic code through nucleotides, which equal a codon, the redundancy, the code is a degenerate, if you will. So what you're seeing is that very complicated process as it goes through. Now, nucleotide sequence divides the coding region. So it designates the beginning, the end of the region, and the end of the region to be translated. You can see that here. Again, same process we've been talking about for quite a while, but what happens is it's going to read that information and create a protein that's going to cause something to happen. The protein will be built in a very specific way. Now, there are 3 reading frames possible, and it kind of depends on the start of the coding region. Correct reading frame is critical. So an incorrect will yield different or likely non-functional proteins. So reading frame number 1 has to fit within a certain criteria. Reading frame number 2, different criteria. Reading frame number 3, different criteria. So you just have to know that there are 3 types of reading frames, and the location in which they start is very important. Now, ribosomes serve as translation machines and really production machines of the protein. The prokaryotic ribosomes comprise those 30-50 units we talked about, creating the 70 S units. They're made from protein and ribosomal RNA, or rRNA. They locate the punctuation sequences on a messenger RNA molecule, and they begin at the start site, which moves along in 5' to 3', like we said several times now. And they maintain correct reading frame, which is important. The lines and forms the peptide bond between the amino acids. So the translation is taking this messenger RNA and translating it to create this protein. The process of gene information in prokaryotes versus eukaryotes, another great video, and you can download that. You can see that on the PowerPoint if you download that and take a look. Now, transfer RNA, tRNAs, delivers the correct amino acid, and each has a specific anticodon sequence. The base pairs with the correct codon, and each carries an appropriate amino acid. After delivery, transfer RNA can be recycled again, so that for an enzyme, the cytoplasm recognizes messenger RNA and attaches the appropriate amino acid. So it can be recycled to some degree. Now, the translation of prokaryotes begins before transcription is complete. So translation of prokaryotes may be starting almost immediately as the transcription is occurring in a much quicker fashion, so to speak. Now, the initiation of translation, part of the ribosome binds to the messenger RNA sequence. It's termed a ribosomal bonding site. The first AUG nucleotide after that site serves as a start codon, completes the ribosomal assembly to start codon. Initiating the transfer RNA brings altered form of methionine, and it occupies that P site we talked about. And we've already seen that on the video if you take a look at that. Now, ribosomal has 2 sites which transfer RNAs combine. The P site, which is occupied by transfer RNA, is carrying the methionine. Another tRNA recognizes the codon in the empty A site, we call that, different sites on this messenger RNA. Occupies the A site and brings the correct amino acid. So this is a process of creating this translation piece to produce the amino acids as we choose. So the amino acids can thus become the protein that we create. Excuse me. The A site and P site are now occupied by the correct transfer RNAs. The enzyme creates a peptide bond between their amino acids. Amino acids from the tRNA and the P site are added to the amino acids from the tRNA at the A site, and they'll ultimately create the given protein that we're trying to produce. There's also a process of elongation of the polypeptide chain. The ribosome advances along the messenger RNA in the 5' to 3' direction, as I said several times, initiating the tRNA exit through the E site, that's the exit site, as it were. And remaining tRNA carrying both amino acids occupies the P site. A site transiently becomes empty. A tRNA then recognizes the codon and A site quickly attaches, and thus we are creating that specific protein downstream. Now, another step is the peptide bond formed between the amino acids. The ribosome advances one codon on the messenger RNA. The tRNA exits the E site. The new tRNA occupies the A site. The process repeats again and again. And once the ribosome clears the initiating sequence, the ribosome combine. The the polyribosome or polysome is then created. Now termination is the last step. Elongation continues till the ribosome reaches the stop codon, if you will, the terminator. It's not recognized by the transfer RNA. Enzymes free the polypeptide. They break the covalent bond joining to the transfer RNA, and thus we have this protein that we've created based on these various amino acids that we've been identified. The freed ribosome falls off the messenger RNA, and falls off the messenger RNA. If the messenger RNA falls off it, I guess we would always say that. It disassociates the component subunits 30 and 50. The subunits can be reused to initiate translation of other sites. So you see that the thing starts all over again at another point. Now, post-transitional modification, the polypeptides often are modified after synthesis. Some must be folded into specific three-dimensional structures with association of protein chaperones. And I'll explain that in a minute. Imagine they may be a primary protein, secondary protein, tertiary protein, or quaternary protein, depending on what we're trying to break, trying to produce, I mean. So the idea is that these proteins will have to be developed in that three-dimensional form in one of those. Now, chaperones are just what they sound like. If you know what chaperones are people that go to dances, if you will, that sort of keep kids from doing things they shouldn't be doing, at least in the dances. And so chaperones are directors. They sort of direct things to happen in a certain way. And it's exactly the same in this post-transitional modification point. Chaperones direct the protein to the right place so it can be modified in some fashion. That modification will probably occur at the Golgi complex somewhere. So the polypeptides are destined for transport to the cytoplasmic membrane or modified with a single sequence. So it may go to the Golgi complex, and ultimately it may be excreted as a toxin, or maybe secreted as a given protein to break down other nutrients. The nutrients can be absorbed by the cell. Lots of different alternatives. There's a series of hydrophobic amino acids, and it tags them for transport as far as that single sequence. The single sequence is moved by proteins in the membrane so that the protein is pure and not modified to make it so it doesn't function as well. Now, the difference between eukaryotic and prokaryotic gene expression is pretty extensive. Eukaryotic translation differs. The messenger RNA is synthesized in a precursor form, pre-messenger RNA, and it must be processed during and after transcription. So the 5' is capped with methylated guanine derivative. It binds specific proteins that stabilizes and enhances the translation. The 3' is modified via polyadenylation, if you will, and it's addition of the 200 adenine derivatives to the new 3' end, and the poly A tail stabilizes the transcript and enhances translation. Just a different way of establishing the start and the stop. And this is the difference between the two, in that messenger RNA is not processed, and there's a cap that we talked about. Messenger RNA does not contain introns, and in eukaryotes, the messenger RNA contains introns which are removed by splicing. In prokaryotes, translation of messenger RNA begins as being transcribed. In the messenger RNA transcription in eukaryotes, it's transported out of the nucleus so it can be translated into cytoplasm, and most likely by the ribosomes. Messenger RNA is often polycistronic. Translation usually occurs at the beginning of the first AUG codon that follows the ribosome binding site. In messenger RNA eukaryotes, it's monocistronic, and the translation begins at the first AUG codon. Now, eukaryotic translation differs in that splicing removes the introns, the non-coding sequence occurs, and exons are expressed in different regions. The messenger RNA transported to cytoplasm. Messenger RNA typically is monocistronic, and the ribosomes are 80S versus the 70S we talked about, made up of 40S and 60S subunits. The important difference for targeting of antibiotics, obviously, we've talked about several times. This is a great look at that process. You can download this again through your PowerPoint out of the D2L module. Sorry. Now, sensing and responding to environmental fluctuations, we have to talk a little bit about that, and that's that microorganisms are constantly facing a changing environment, and to survive, they have to adapt rather quickly. For instance, E. coli and mammalian intestinal tract in the human gut, there's alternating periods of feast and famine where we're eating and not eating. The host eats, bacteria take up the amino acids, vitamins, nutrients, and it shuts down the biosynthetic pathways and channels energy in the production of cellular components, for example, DNA, protein. Between meals, the nutrients are depleted, or that's the fasting stage, as it were. And the bacteria biosynthetic pathways are activated, the energy is consumed, growth is slowed. As the animal defecates and the human defecates, some E. coli is excreted, and they face an entirely different set of conditions, obviously. So that just kind of explains how they adapt to the different environment. Now, since they respond to environmental fluctuations, we have what's called signal transduction, and that transmits information from outside the cell to inside the cell and allows the cell to monitor and react. Sort of think of this as the thermometer or the thermostat that says, okay, you go down here, you're going to have to heat things up inside. That's kind of the way that signal transduction works. With quorum sensing, some organisms can sense the density of their population, and they can see how many you have around them. It allows cells to activate genes useful with critical mass. So basically, how to live within this spectrum where the nutrients may be decreasing and the waste products may be increasing. And so, for example, a biofilm formation occurs, the pathogens and infective processes may change, and some can detect interfere with signaling molecules of other species. They can eavesdrop and obstruct conversations even. So that's just some of the interesting things that our prokaryotic friends can do. Now, there are two component regulatory systems that help the sensor respond to environmental fluctuation. One is a membrane spanning sensor, and it's a modified internal region response to specific environmental variations, and it phosphorylates amino acids and messages at work. There's a response regulator, and it's a phosphate group that's transferred from the sensor, and the regulator turns the genes on or off in that response. Examples include E. coli using nitrite as a terminal electron acceptor, the pathogen sensing magnesium levels to recognize it within a host cell, so they are able to use that as a mechanism. Good regulatory protein video you can see right here. Now, natural selection can play a role in genetic expression, obviously. Excuse me. Expression of some genes changes randomly in cells. There's a mutation aspect that's very high. It enhances survival, at least part of the population, so they can react to strong changes in the environment. And antigenic variation is really alteration of characteristics of surface proteins. It allows the pathogens to stay one step ahead of their host defenses, so to speak. For example, Neisseria gonorrhea has many genes for pilin, if you will, protein subunit pilin. They're only expressed as a gene in expression locus. It randomly moves genes in and out of the expression locus, and the immune system responds initially to dominate the pilin type. The bacteria that have switched the types that survive. So they kind of switch the message, so to speak. The phase variation involves switching genes on or off, allows E. coli to attack via pili and then detach by turning them off. So different techniques. We look at bacterial gene regulation. The genes can be treated, expressed, or regulated. A set of regulated genes transcribed as a single messenger RNA, along with its control sequence, is termed an operon. And for example, a LAC operon for lactose metabolism, LAC operon, lactose operon. And so the separate operons are controlled by a single regulatory mechanism, which constitutes the regulon, and the global control is simultaneous regulation of these numerous genes. So there's a regulation of all these things that are happening at a single control point. And this looks at the LAC operon, the combination of switches. Great video to download and take a look at. Now, enzymes can be grouped by type of regulation, and they can be constitutive enzymes, inducible enzymes, or repressible enzymes. Constitutive enzymes are synthesized constantly. They're typically indispensable roles in the central metabolism, for example, enzymes in glucose and break down glucose. Inducible enzymes are not routinely produced. They synthesize only when needed. For instance, beta-galactidase is turned on only when lactose is present and avoids the waste of resources. And repressible enzymes are produced routinely. They're turned off when not required, for example, antibiotic pathways such as amino acid synthesis, but they are only turned off when they are not needed specifically. Now, bacterial gene regulation, there's mechanisms to control gene transcription, and that must be really reversible, allow cells to control relative number of transcripts produced. So the two most common are alternative sigma factors and the DNA binding proteins. The first, the alternative sigma factors. The standard sigma factor is a loose component of RNA polymerase that recognizes specific promoters for genes expressed during the routine growth conditions. Alternative sigma factors recognize different sets of promoters for expression of specific groups of genes. For example, sporulization of Bacillus subtilis controlled by multiple different alternative sigma factors. Now, the mechanisms to control transcription of DNA binding proteins can act as repressors or activators. So repressors block the transcription, giving you a negative regulation. Binds to operator, stops the RNA polymerase. Repressors are allosteric. That means they have binding sites and alter the ability to bind to the DNA. There are two general mechanisms, induction and repression. Induction is the repressor binds, blocks the transcription. The inducer binds the repressor is unable to bind. The repression is the repressor unable to bind to DNA. The core repressor, if you will, attaches the repressor. The complex now binds to DNA and blocks the transcription. So two mechanisms we have to control that. Great look at induction in the video if you want to take a look at that. DNA binding proteins and repressors. This walks through very much the same thing we talked about. I'll let you look at this and you kind of walk through it with the information we had. I'm not going to go through it again, but you get the idea. Now, DNA binding proteins can act as repressors or activators. If they're activators, they facilitate transcription. They are positive regulation. They have an ineffective promoter preceded by an activator binding site, and the binding of activator enhances the ability of RNA to initiate transcription of the promoter. The inducer binding to the activator allows the binding of DNA, and this takes this process. As you can see, the RNA polymerase cannot bind to the promoter unless the activator is bound to the activator binding site, but the activator is in an inactive form. In this case, what happens is the inducer binds to the activator and changes its shape, allowing the activator to bind to the site. RNA polymerase can then bind to the promoter and initiate that transcription as we discussed. Transcription factors, another great video. Now, the lac operon is a model of lactose and the lac operon. No lactose, the repressor prevents the transcription. Lactose present, some is converted to inducer allolactose. So no lactose, there's no repressor created. Lactose, some is converted to this repressor. It binds the repressor releases the operator, RNA polymerase induces the operon. It occurs only when glucose is unavailable. So that's when we're using lactose as opposed to glucose. Galactose and the lac operon continued. Carbon catabolite repressor, CCR, prevents expression of lactose in the presence of glucose. So they prioritize the carbon energy sources and yields what's called the dioxic growth. The glucose transport system, such as glucose, the catabolic activator protein, CAP, is required for transcription, and the functional only when bound by an inducer, cyclic AMP. Cyclic AMP is made when glucose is low. The inducer exclusion, the lactose transporter, is blocked during the glucose transport. So this takes you to the time of incubation hours, as it were, and you see how that all works between the glucose and lactose. Now this takes you through another model, as it were, step by step. And again, I'll let you look through this. I won't go through this in this recorded lecture because it'll make this lecture so much longer, but you can follow these step by step and see the information we've already discussed. Now eukaryotic gene regulation is a little different. It's far more complicated. And there's a variety of approaches. There's modification of the chromosomal structure. There's regulation of initiation of transcription. There's altering the pre-messenger RNA processing modification. There's RNA interference. There's all sorts of different processes in order to manage that. RNA interference, RNAi, is a short RNA strands which join the multi-protein unit. And the RNA-induced silencing complex are at risk, if you will. The RNA strand serves as a probe for binding to the messenger RNA. It tags the messenger RNA for destruction. Risk is catalytic and rapidly silences the transcript. So this is a way to stop the process of messenger RNA. It destructs it or degrades it. MicroRNA and short interfering RNA are about 24 nucleotides and are produced very differently throughout the system. Now, let's look at genomics, if you were. Let's look at Haemophilus influenza, which is the first genome published in 1995. Since then, microbial genome sequencing is pretty common. There have been hundreds of discoveries, and the data interpretation is complex. There's a promoter orientation, which DNA strand is template, and reading a frame. So the new field of bioinformatics has emerged from that process of learning that genomics. So analyzing a prokaryotic DNA sequence, there's a positive strand. It's used to represent the sequence of the RNA transcript. For example, ATG in positive DNA indicates a possible start codon. Computer search for the open reading frames, and the stretches of nucleotides generally longer than 300 BP. They begin with a start codon and end with a stop codon we talked about before. The ORF, the open reading frame, is potentially encodes the protein. Other characteristics, for example, upstream sequence serving as ribosomal binding site, also suggests that the open reading frame encodes the protein. These can be compared with published sequences or databases that include the sequences, and it can identify the presumed function of that given set of genes, if you will. Now, metagenomics is the analysis of total microbial genomes in an environment. They can study all microorganisms and viruses in a community, and it's not limited to just those that grow in a culture. They can track changes in composition of microbiota of individual over time, healthy and diseased, if you will. They compare microbiota at different body sites. They also compare between different individuals. It allows them to study microbial life in oceans and soils, and it gives a new understanding of the extent of biodiversity. It may lead to discoveries of useful compounds, for example, antibiotics, and there's a tremendous amount of data which presents challenges. It's interesting this book still talks about antibiotics as the center core of our defense against disease. Primarily, as far as the study and research that's going on now, we're moving away from antibiotics, which kind of a dead end, in the sense that we have a bacteria, we create an antibiotic against it, the bacteria develops a resistance, we create another bacteria against it, and the bacteria becomes more stronger and stronger, more resistant, and the antibiotics become more destructive to human cells. They become less therapeutic. And so the use of antibiotics is kind of moving away from the techniques we've had in the past. We're moving towards vaccinations and gene reconstruction, if you will. So there's a tremendous amount of data that's present in these challenges, but the book sort of suggests that those are to make new antibiotics, and the truth is that's not where we're going at all. So this is a look, if you will, at the blueprint of life from DNA and protein. Have a good day.